Hello everyone, in this video I'm going to speak about X inactivation, which is a really interesting topic. In this video I'm going to speak everything about X inactivation. What is X inactivation? When does it happen? How does it happen? I'm going to speak about the full mechanism, and I'm going to speak about the euchromatin and heterochromatin. So watch till the end and enjoy the video. So first, let's speak about what is X inactivation. So you know that in normal cells, the females have XX sex chromosomes, and the males have XY sex chromosomes. And since the X chromosome is a very important one, and it carries not only sex genes or sex traits, but also somatic traits. So X chromosome is very important. And to prevent imbalance between female and male, since female has two X chromosomes, so in each cell, one of the two X chromosomes is inactivated. It turns inactive. So in this case, we call the active one XA and the other one XI, the inactive one. Now, which chromosome is inactivated? You know that in the cell there is like one father's chromosome and one mother's X chromosome. Which one is inactivated? The chance is really 50%. So it can be either of these. When does the X inactivation happens? Actually, X inactivation happens in the embryonic stage. So when the cells are undifferentiated, before cell differentiation. So let's say that this is the fertilized oocyte. The fertilized oocyte is divided into 2, and then into 4, and then into 8. When the embryo is in the 8 cell stage, X inactivation starts. So each one of the cells, in each one of the cells, one of the X's are inactivated. And as I told you before, the chance is 50%. So it can either be the mother's one or the father's one, which is inactivated. So this is the embryo, this is the embryonic stage. In each one of the cells, either the father's X chromosome is inactivated or the mother's one. And as I told you before, the chance is 50%. Afterward, there is the differentiation stage. So in the differentiation stage, all the cells which are derived from a mother cell in which the father's X chromosome is inactivated, they have father's X chromosome inactivated. And all the cells which come from a mother cell in which the mother's X chromosome is inactivated, they have all a mother's X chromosome inactivated. Afterward, in the female body, 50% of the cell will be mother's X chromosome inactivated, and 50% of them will be father's X chromosome inactivated. The most known example of this is what's called the calico cat. You see, the calico cat has some orange fur and some black fur. And the reason for this is that, so the calico cat is always a female. You should know this. And the fur color trait is on the X chromosome. So let's say we have a cat, we have a male cat, okay? Let's say first we have a male cat. In the male cat, there is only one X. So this X could be either black, and then the cat will be totally black, or orange, and in this case, the cat will be totally orange. But in case of female, we have 3 choices. So the cat, either both of them will be black and then the cat will be totally black, or both of them will be orange and then the cat will be orange, and then, or one orange and one black, and then the cat will be like this. Why? Because in the orange, when the fur is orange, then the black X chromosome is inactivated. And when the orange X chromosome is inactivated, we see some black patches. So because, as I told you, the fur color trait is on the X chromosome. Of course, you're seeing some white patches here, and in these patches, this trait, the fur color trait, is not even expressed because of some epigenetics reasons. But this is in general, okay? Now, let's speak about the mechanism. The mechanism of X inactivation. How does X inactivation occurs? So this is an X chromosome. Okay, this is the short arm, this is the long arm, and this is the centromere. In this position, there is something called the XIC, which is the X inactivation center. It exists on the XQ13 position. So on the long arm, the Q, and the 13 position. In this region, there are several genes, but the most important gene, which is impacted directly in X inactivation, is the XIST. The XIST is the X inactivation specific transcript. This gene is implicated, as I told you, directly in the X inactivation. Now, you know that the chromosome is double-stranded DNA. So on one strand, we have the XIST. On the other strand of the DNA, we have the T6. The T6 is exactly the antisense of XIST. Even from the name, T6 is the opposite of XIST. If you read it from right to left, it's T6, because T6 is the antisense of the XIST, which occurs on the other strand of DNA. And like the T6 plays the principal role in X inactivation, while the T6 plays an opposite role, and T6 overlaps the role of XIST. There is also another region called the X control element. I'm going to speak about this in details in few minutes, by the way. There is another region here called the X control element, XCE, which is believed to play a very important role in managing the X inactivation. Now, this is the X chromosome. These are the double-stranded DNA. On one strand, we have the XIST. On the other strand, we have the T6, which is transcribed in the other direction. Now, after on the downstream of T6, we have the XCE, the X inactivation controlling element. This is the other X chromosome. Now, first, in the early embryonic stage, both X chromosomes express the XIST, but the expression or the transcription of XIST start from a primer called the P0. You're seeing this is the primer. It's called the P0. The RNA you're seeing here is a non-coding RNA. What does non-coding RNA means? This RNA is not translated to a protein. This RNA plays as an RNA. It's not translated into a protein. Okay. The RNA which is transcribed from the P0 is an unstable RNA. And because of this, it does not accumulate. Now, afterward, there is a factor, which is a blocking factor, produced in the cell. And this you should know very well. The blocking factor is produced in a very specific amount, which is sufficient to block only one chromosome. This is the blocking factor. The blocking factor is, I will repeat it because this is very important. The blocking factor is produced in a very in a very specific amount, which is sufficient to produce sufficient to block only one chromosome. The blocking factor blocks the XIST, and when the XIST is blocked, then this chromosome cannot be inactivated anymore. Why the blocking factor blocks only one chromosome? Because we need like this chromosome is going to be active. And in each cell, we need one active chromosome. And for the chromosome to be active, the exist should be blocked. I don't know if you understand this. So let's say if the female is triple X, XXX, then we will have a blocking factor sufficient to block one X, and then one X will be will be active, and then the other two X's will be inactivated. Let's say if the female is like 4 X's, then 3 X's will be inactivated and 1 X will be active, and so on. So the blocking factor is always sufficient to block only one X, and then this X is going to be active because the exist is blocked. On the other X, which is going to be the inactivated one, the transcription of the exist moves from the primer P0 to another primer called the P1, or there is a new primer called P2. So the transcription here takes place either from P1 or from P2. And when the exist is transcribed from P1 or P2, then the RNA is going to be stable and is going to accumulate in the cell. And this is what we call the stable exist RNA. And this RNA is the principal player in the X inactivation process. Okay, so this is an unstable RNA. The blocking factor is sufficient to block only one X, which is going to be the active X. So this is the active X, and this is the inactive X chromosome, in which the exist is transcribed from P1 or P2 primers. Now let's see what happens. So this is the X chromosome. This is the exist in the XIC center or the XIC region. The exist is transcribed to the stable RNA, and then the stable RNA is going to accumulate in the cell, and it's going to cover the X chromosome like this. In this case, many modifications, I'm going to speak into details, but now I'm speaking in general. So some modifications are going to happen in the X chromosome, and then the X chromosome is going to be compacted. Let's see how. So this is the RNA, the stable RNA. This is the X chromosome, the inactive one, the inactivated one. Now, let's first see what is euchromatin and heterochromatin means. What does it mean? So euchromatin is a structure of the chromosome. Euchromatin is the structure of almost all the somatic chromosomes. So almost all the somatic chromosomes are in the euchromatin structure, or have the euchromatin structure. The euchromatin structure is 8 histones. So these the purple circles you're seeing here are histones. These are proteins called histones. So each 8 histones are compacted together, and the DNA is like rolling around the 8 histones. For the 8 histones, we have H2A, H2B, and H3, H4, and another time H2A, H2B, H3, and H4. Then we have another group and another group and another group. This group we call the nucleosome, which consists of 8 histone molecules and 146 base pairs of DNA. This is the euchromatin. So normally the chromosome is like this. This is the DNA. Now we have 8 histones with the DNA around it, and then they coil together to form the chromosome. This is the euchromatin structure. What happens in X inactivation is that the chromosome is converted from the euchromatin structure to the heterochromatin structure, which is more condensed. Let's see how. So You should know that the histones have tails, histone tail. And these tails are amino acids because you know the histones are proteins and then the tails are like amino acids. These tails can be modified, like there can be histone tail modification, can be methylation, acetylation, phosphorylation, or ubiquitination. So the tails can be modified. What happens in X inactive, so what we are interested in is methylation and acetylation, most importantly here. When the tails are acetylated, what happens? Let's see what happens in case of acetylation. In case of acetylation, the nucleosome structure is relaxed. It's like this. So this is the nucleosome structure. In case of acetylation, this structure is relaxed, and then the histones are separated from each other, and then the DNA is relaxed. This happens in case when the DNA wants to be copied. So when the DNA wants to be copied, this facilitates DNA copying. So what happens is acetylation to the tails of histones. Now, the opposite is in case of methylation. So what happens in case of methylation is that the nucleosomes are compacted together more and more. So this is what happens in case of methylation. So the histones are compacted together. Okay, this is in case of methylation. And this is what happens in case of X inactivation. In case of X inactivation, this is what happens in case of X inactivation. The histones are, and so the DNA is compacted together. So epigenetic modifications that happen in case of X inactivation. So when XIST is transcribed, and when we have the stable RNA, at the 8-cell stage, at the embryonic stage, when the embryo is at the 8-cell stage, there is H3K4 methylation. K4 is the lysine number 4. So K is the abbreviation for the amino acid lysine. So on the histone 3, we have methylation on K4, on the histone 3 tail. We have methylation on the K4, and then we have deacetylation. You know, I told you before that acetylation relaxes, deacetylation increases the compaction. Then at the 16-cell stage, we have also methylation on histone 3 lysine number 27. And this happens because of a protein called the PRC2. This protein is recruited by the stable RNA, the XIST RNA I told you about. So the XIST RNA recruits proteins to undergo these methylation and deacetylation modifications. Now in this stage, there is one protein called the H2A, this protein, this histone. This histone is substituted with another histone called the macro H2A. And in this stage, the process becomes irreversible. Why? Because the macro H2A inhibits any remodeling of the chromosome. If there is any chance for remodeling before this stage, there is chance of remodeling. Now, after the H2A is substituted by the macro H2A, there is no chance for remodeling, and the process becomes irreversible. Afterward, in the 32-cell stage, there is also methylation on the H3K9. After all these modifications, the chromosome becomes so compacted together. So this is the normal X chromosome, and then when they exist in the XIC start to produce the stable RNA, the RNA covers the X chromosome. In this case, the X becomes the inactivated X chromosome, and then after all these modifications, the modification I told you about, the X chromosome is tightly compacted together, and it becomes a Barr body. This is what we call the Barr body. So the X chromosome is compacted into a Barr body because of the methylation and the deacetylation of the histone tails. Now these are some images to see the Barr body. The Barr body is always on the edge of the nucleus. This is a nucleus. Everything you're seeing here are euchromatins, or everything you're seeing here, they are the euchromatin. They are relaxed. They are the other 45 chromosomes. They are relaxed. But what you're seeing here is the compacted Barr body. Now, this is everything I wanted to tell you about X inactivation. I hope you enjoyed this video. If you really enjoyed it, like and share. If you have any questions, write the question in the comments. If you have any suggestions for other topics I can speak about, please write in the comments also. And see you in the next video. Bye.